guanine, and thymine. And these nucleotides pair complementary. So adenosine always pairs with thymine, and cytosine always pairs with guanine. If we were to look at the DNA molecule, recognize that it actually is in a double helix. If I can do this here. Meaning that it kind of looks like this, coiled in a double helix. Think of a ladder then that's just been twisted. So if we untwist that ladder, what we see is a molecule that's composed of two strands with the complementary base pairs like so. And so if we were to put our complementary pairs here, everybody in agreement with what I have on the board? So I've got my DNA molecule. It's two complementary strands of nucleotides. And so this is our sequence of DNA. Now, what's great about this molecule is that it's semi-conserved, and when it goes to replicate, it essentially will unzip and replace the missing nucleotide with a complementary nucleotide, so that you end up with one strand that's composed of an old strand of nucleotide, or I should say one DNA molecule with an old strand of the nucleotide sequence and a new strand of the nucleotide sequence. Can you distinguish those two colors?
No.
And so the other strand will be just the reverse then, consisting of the same sequence, but a different new strand. So in replication, we end up with two DNA molecules identical. The sequence is identical to the original molecule. Now, with bacteria, their genome is circular. So if we were to look at a bacterial genome, even though we might represent it as being a bunch of squiggly lines like this in the cell, because it's really a big genome. When we go to draw it, we can draw it as being circular. Like so. And now I'm representing one strand of the DNA molecule on the outside, one strand of the DNA molecule on the inside. Okay. The other thing that the DNA molecule does is it codes for messenger RNA, or for RNA in general, ribonucleic acid. Now this molecule here has a number of roles, and there are a number of types of RNA, messenger RNA, transferrin RNA, ribosomal RNA, and we are still discovering RNA molecules that we're not quite sure what role they're playing. This molecule here reads the message that the DNA molecule has and transcribes it, if you will, into a protein, so that we can then do work with that protein. We've got to take this DNA message and synthesize proteins from it. The proteins are what do the work in our cells, the enzymes and the structural proteins, and they're all coming from this sequence here. Now, the way we produce a messenger RNA, again, is the sequence will unzip, but only 1/2 of the strand will be copied. So if this unzips, maybe the RNA, or the messenger RNA, will read this part of the sequence. Or maybe it'll read this part of a sequence. It depends on where a motor establishes where the reading should take place. And that is more than we need to get into. But the cell will unzip, and then the messenger RNA will make a complementary strand from the unzipped portion here. So if we were to pull this away now, so we've unzipped our DNA molecule. Our RNA is going to come in and we're going to replace those nucleotides that have been removed with complementary nucleotides, except for with, in the case of adenosine, RNA uses a uracil molecule instead of a thymine molecule. And it's how we know we're talking about an RNA molecule. It'll have uracil in it. So if we were to look at this sequence here, it would be U, A, G, C, U again, A, G, C, U again, A here. So an RNA molecule is a complementary sequence to the DNA molecule, except uracil is used instead of thymine. Now this RNA molecule will leave the nucleus, make its way out into the cytoplasm of the cell, where it will hook up with a ribosome, and as it moves through this ribosome, it'll be translated. What does it mean to be translated? It means that this sequence here will be converted to an amino acid sequence. Out there in the cell are transfer RNA molecules. And these transfer RNA molecules are specific to certain amino acids. We have 20 different amino acids, so there are 20 different transfer RNA molecules in your cell out there picking up amino acids. They bring those amino acids back to the ribosome, and when their sequence matches a sequence on the messenger RNA sequence, then they'll drop off their amino acid, building a chain of amino acids. This chain of amino acids that's coming from this messenger RNA sequence will eventually fold up on itself and then either become structural or enzymatic. Being active will cause reactions to take place, or if it's structural, will be used in the biosynthesis of macromolecules. The DNA sequence is a triplet code, so every three nucleotides codes for an amino acid. This codes for a specific amino acid. This codes for a specific amino acid. This codes for a specific amino acid. So eventually you get a chain of amino acids that then will fold into a protein. That's a very quick and dirty review of genetics. Again, I would recommend that if you're not comfortable with it, that you review chapter 7. Okay, so when we think about bacteria, one of the things that you think about is the diversity of bacteria. Simply put, they have to be able to adapt quickly to a changing environment. And so this means a lot of times that they have to change the expression of genes. When I talk about expression of genes, I mean the genes that then are producing protein. So if you have a protein that is necessary, an enzyme is necessary for the breakdown of glucose, but you can also break down lactose, and you find you're in an environment where there's no glucose, you need to change your gene expression so that you can now produce an enzyme that will break down lactose. And so bacteria have that ability to a certain extent that they can, some of them can change their gene expression based on the environment that they find themselves. In addition to that, because of their generation time, and we've talked about this here, some bacteria reproduce every 20 minutes. Because of their generation time, mutations that take place or changes in the nucleotide sequence can happen fairly quickly in a population that would create maybe a new protein or a new enzyme that might give an advantage to that cell. We're going to see a study that does just that. Not this time, but the next period we'll open up with a study by Richard Linsky that actually looks at some of the evolutionary changes that took place in E. coli. The other thing that we can say about bacterial genetics, the microbes themselves are haploid. I've erased it, I guess. A single circular genome, okay? Not several chromosomes like we have, but a single circular genome. So how can changes in the genome take place? Two ways. Mutations and gene transfer. With mutations, we are actually talking about changes that take place in the nucleotide sequence. And then those changes, of course, would be passed on to each and every generation after that. With gene transfer, we're talking about picking up genes from the environment, or from viruses, or from some other microbe. This type of genetic change is going to be vertical, and this type of change is going to be horizontal. This is going to be from mother cell to daughter cells. This is going to be from one microbe to another microbe. In talking about gene mutations, if a gene happens and if a mutation happens and the properties of the gene are not changed, so the enzyme doesn't change, the gene didn't change anything, that is referred to as a wild type mutation, meaning that there's no difference in the mutational microbe and the one that you would see in the wild. The mutation had no effect. This is also referred to sometimes as a silent mutation.
So the wild type is the silent mutation?
Sometimes referred to as a silent mutation.
And that's...
See, a wild type mutation you don't see. It looks like the microbe in the wild. A silent mutation also is not observable. Now, mutations are spontaneous. And each gene mutates. So all your genes mutate, all of the bacterial genes mutate, but the rate of mutation can vary. Some might mutate every 10 to the minus 4. Some might mutate every 10 to the minus 12 generation. So you can see wide frequency of mutations. So we're saying that this would happen. In this case here, you would get a mutation once every 10,000 cells. In this case here, you might get a mutation once every 1 trillion divisions or cells. So it really can vary depending on the gene. Some genes mutate more frequently than other genes. And then our cells have many repair mechanisms as well. So a lot of times a mutation might take place that gets fixed. The other thing that we can say is that genes mutate independently of each other. So if one gene mutates, say, every 10 to the 4th, and another mutates every 10 to the 6th, if you wanted to know when you would see both mutations, you would add those together, and you would get a mutation of both of those occurring every 10 to the 10th. Let's talk about base substitutions. This is what happens when one of the nucleotide bases gets changed. So during DNA synthesis, during the replication of the DNA molecule, an incorrect nucleotide gets put in place. A type of base substitution is a point mutation. So a very specific base pair is changed, and this can cause a silent mutation, as we've already mentioned. It can cause a missense mutation. So let's go back to this sequence here.